Hey guys, welcome and welcome back to my channel. This is Dr. Prasanjit Das, currently JR1 in the Department of Pharmacology, AIMS Kalyani, and I am back with one of my seminars. So, without wasting any time further, let's get into today's topic. So today I am going to discuss about the topic that is pharmacogenetics and its therapeutic implication in drug metabolism. So it is one of my seminar topics, and under this we are going to discuss this topic under the following headings, that is the history of pharmacogenetics, then the drug metabolism, that is the different phases of drug metabolism and their significance, then the therapeutic implications of pharmacogenetics and pharmacogenomics. So coming to the first slide, that is the history of pharmacogenetics. So before we go into the history of pharmacogenetics, let's first discuss what is the definition of pharmacogenetics and pharmacogenomics. So pharmacogenetics is the study of the genetic basis for variation in drug response and often implies large effects or small number of DNA variants, that is small number of DNA variants. Whereas pharmacogenomics, on the other hand, studies large numbers of variants in an individual or across a population to explain the genetic influences of drug response. So basically in pharmacogenetics we are going to study small number of DNA variants, whereas in case of pharmacogenomics we are going to study large number of DNA variants and how it is actually affecting the pharmacological effect of a particular drug. So we have several examples where a particular drug is showing different effects in the different individual. So basically a single drug can produce different types of effects in different individuals, and behind that there are several factors which are going to actually affect it, like the environmental factors, then we have the drug factors, and then we have human factors like the genetic factors, etc. So the key genes which are actually known to be involved in driving variable drug actions in different individuals are called as pharmacogenes. Okay, so pharmacogenes may influence the drug response via variable drug concentrations, that is it can either affect the pharmacokinetics, or variable drug effects can be seen, that is it can either affect the pharmacodynamics. So it can affect either the pharmacokinetic property of the drugs or it can affect the pharmacodynamic property of the drugs. So pharmacogenes include those genes which are actually coding for several enzymes, that is the drug metabolizing enzymes. Then these genes also code for several drug transport molecules, and drug targets. So as you can see, drug metabolizing enzymes, it is affecting the drug metabolism, drug transport. It helps in the transport of the drugs, that is the molecules which are responsible for the distribution, absorption, etc. Then we have the drug targets which are influencing the pharmacodynamic property of the drug, that is the effect of the drug. So all in all, pharmacogenes are affecting the pharmacokinetics and the pharmacodynamics of the drug. Now coming to some of the first examples of genetically determined variable drug response due to the pharmacokinetic variability. So first is the case of succinylcholine. So succinylcholine can lead to succinylcholine apnea because of the prolonged paralysis of the respiratory muscles. It occurs because of the presence of atypical pseudocholinesterase, because there can be pseudocholinesterase deficiency, and as a result this leads to succinylcholine apnea, that is succinylcholine is not metabolized properly, leading to prolonged paralysis of the respiratory muscles. Next example is isoniazid induced hepatotoxicity. Isoniazid is used as an anti-tubercular drug and it can cause hepatotoxicity in case there is deficiency of N-acetyltransferase. So these are some of the examples where the drug response is actually affected by the genetics. And now coming to the father of pharmacogenetics. So Arno Motulsky, as you can see in this image, is considered as the father of pharmacogenetics. He defined the field of pharmacogenetics and he had broad interest basically in G6PD deficiency. So G6PD deficiency, as you can see here, if the RBC is having G6PD deficiency, then the reactive oxygen species will cause the damage to the erythrocyte, and as a result the lifespan of that particular erythrocyte will decrease. So this is the case of G6PD deficiency, and this is one of the areas where Arno Motulsky have worked initially. So and it was his field of interest. Now coming to the next slide. So drug metabolism. So what happens in drug metabolism? So basically the drugs which we take in, that is the drugs which are used for pharmacological purpose, are actually hydrophobic in nature. Okay, hydrophobic in nature means it repels the water. So by drug metabolism, this hydrophobic drug is converted into a hydrophilic metabolite which can be easily excreted in urine or can be excreted via bile. So if the drug is having a high molecular weight, then it will be mainly excreted via the bile in the intestine and in feces. So this is the case of the drug metabolism. So drug metabolism is done basically by the drug metabolizing enzymes. Initially these enzymes were known as drug metabolizing enzymes, but now they are known as xenobiotic metabolizing enzymes. This is because these enzymes not only metabolize the drugs, but they also metabolize any other substance which is foreign to the body. That is, any substance which is foreign to the body is known as xenobiotic. So they are now aptly termed as xenobiotic metabolizing enzymes. And these enzymes will be also responsible for metabolizing some of the endogenous substances also. Now coming to the phases of drug metabolism. So we have phase 1 reactions and then we have the phase 2 reactions. So phase 1 reactions include the reactions like oxidation, reduction and hydrolytic reactions, whereas phase 2 reactions are the reactions in which the enzyme are present that catalyze the conjugation of the substrate, that is conjugation of the substrate, that is the phase 1 product. Means phase 1 will produce a particular product which will become the substrate for the phase 2 reaction, and in that case there will be conjugation of a particular molecule to this phase 1 product. So this is the case of drug metabolism. So in phase 1 we have enzymes which cause the introduction of several functional groups like hydroxyl group, then we have carboxylic acid group, then we have SH group, that is the thiol group, then we have the amino group, and then we have the ether group also. So these are the different groups which can be introduced with the help of the phase 1 reaction. The main purpose of phase 1 reaction is that it leads to inactivation of the drug. So this is the main purpose of phase 1 reaction. So this inactivation is seen in case of most of the drugs, but there are several group of drugs which are undergoing phase 1 reactions, and after phase 1 reaction they get activated to their active metabolite. So these kind of drugs are known as prodrugs because these are actually getting activated with the help of phase 1 reaction. So examples of those drugs which are getting activated to their active metabolite form because of the phase 1 reactions includes clopidogrel, then we have cyclophosphamide. All these drugs are prodrugs and they are converted to their active metabolite with the help of enzymes, that is CYP enzymes. So we will be coming to the CYP enzymes in this slide only. So here you can see one example of the drug which is undergoing both phase 1 and phase 2 reactions. So we have the example of phenytoin, which is highly lipophilic, that is hydrophobic in nature. So it undergoes the phase 1 reaction with the help of the cytochrome enzymes, that is CYP enzyme, and it is getting converted into 4-hydroxyphenytoin, which is slightly soluble in water. Then it undergoes the phase 2 reaction where basically it is undergoing a process called as glucuronidation, that is with the help of UDP, glucuronyl transferase, and this makes it completely water soluble in nature, and as a result it can be easily excreted out. So this is the complete mechanism, that is phase 1 basically does the inactivation of the drug and phase 2 makes that inactivated product even more water soluble. So this is the main purpose of phase 1 and phase 2 reactions. Now coming to different enzymes which are responsible for catalyzing the phase 1 reactions and phase 2 reactions. So first, phase 1 enzymes. In phase 1 enzymes are the enzymes like cytochrome P450 enzyme, which is the major group of enzyme. Then we have FMOs, that is flavin containing monooxygenase. Then we have EH, that is epoxide hydroxylase, so it causes the hydrolysis of the epoxide groups. So these enzymes are responsible for causing the inactivation of the drugs, and in case of prodrugs they cause the activation of the drug. Next is phase 2 enzymes are mainly the transferases, and they are responsible for increasing the water solubility of the drug. As a result, it can be easily excreted from the body. So under phase 2 we are having several enzymes like sulfotransferases. So sulfotransferases, from the name it is clear, it causes the addition of the sulfate group. Next is UDP glucuronosyl transferase. As I have already mentioned here, UDP glucuronosyl transferase, that is UGT. It is used for addition of the glucuronic acid. Next is glutathione S transferases, which causes the addition of the glutathione group. Then N-acetyl transferases, which helps in the addition of the acetyl group, and methyl transferases, which helps in the addition of the methyl group. So all these enzymes are called as xenobiotic metabolizing enzymes togetherly. So this was about the phase 1 and phase 2 enzymes and the main purpose of phase 1 reaction of drug metabolism and the main purpose of phase 2 reaction of the drug metabolism. Two key points which we need to remember is in phase 1 it mainly causes the inactivation of the drug, an exception is prodrug, and in phase 2 it mainly causes the increase in the water solubility of the drug so that it can be easily excreted out of the body. Now coming to the different phase 1 reactions. So here you can see the different phase 1 reactions. It includes oxidative reactions, then we have hydrolysis reactions, and different phase 2 reactions are mainly the conjugation reactions. So we will take some example of the drugs. So under oxidative reaction we have N-dealkylation. It is seen in case of drugs like diazepam, erythromycin, tamoxifen and theophylline. So basically we have to remember these drugs. There is no way to actually understand here. It is basically to by heart the things. So next we have O-dealkylation, which is seen in case of indomethacin and dextromethorphan. Then in case of aliphatic hydroxylation, the drugs which are involved are ibuprofen, then phenobarbital, then we have cyclosporin. Then next is aromatic hydroxylation. Aromatic hydroxylation occurs in case of phenytoin, in case of phenobarbital, in case of propranolol, also in case of warfarin. Next is N-oxidation, which occurs in case of dapsone, and chlorpheniramine S-oxidation, which occurs in case of simididine and in case of chlorpromazine, omeprazole. Next is deamination, that is the removal of the amino group. It is seen in case of diazepam and amphetamine. Now coming to the hydrolysis reactions. So hydrolysis reaction is basically meant for the drugs like carbamazepine. Then we have some of the anesthetics like procaine. Then we have aspirin, that is cocaine. Then we have lidocaine, procainamide, indomethacin. Next, coming to the phase 2 reactions, that is phase 2 reactions are basically the conjugation reactions. That is glucuronidation. So glucuronidation is done in case of acetaminophen. This is very important. That is paracetamol. So we need to remember this. Then we have morphine, oxazepam, lorazepam. Then we have sulfation in case of methyl dopa steroid and also in case of acetaminophen. But majorly acetaminophen is undergoing the glucuronidation reaction as the phase 2 reaction. Next is acetylation in case of sulfonamides, in case of isoniazid. So isoniazid we have to remember, as I have already mentioned, in isoniazid we are having slow acetylators and the fast acetylators. So it is one of the starting drugs, like the drugs which actually made us think about the possibility of pharmacogenetics, that is the genetic factors involving and influencing the drug response. Next is methylation in case of L-dopa, in case of mercaptopurine, in case of captopril. Then glutathionylation in case of busulfan and fosfomycin. So these are the examples of the drugs which are metabolized by the particular phase 1 and the phase 2 reactions. Now coming to the next slide. So here you can see the different enzymes which are responsible for causing the phase 1 metabolism and the phase 2 metabolism. So in phase 1 metabolism, as you can see here, CYP3A4/5 is the cytochrome enzyme which is responsible for causing maximum number of the phase 1 reactions, and it is followed by CYP2D6, and then it is followed by CYP2C8/9. So these are the three most commonly implicated phase 1 enzymes. Apart from this, we have other phase 1 enzymes also, like we have esterases, then we have epoxide hydrolysis also. Next, coming to the phase 2 enzymes. So in phase 2 enzymes, you can see here the most commonly involved enzyme is UGT, that is helpful in glucuronidation. Then following that we have SULTS, that is sulfotransferase. So it is basically used for the purpose of sulfation. Now coming to the nomenclature of this cytochrome P450 enzyme, as you can see here, it is CYP3A4. So this 3 indicates the family. Then we have A, which indicates the subfamily of the cytochrome enzyme, and 4, it is indicating the specific gene number. So basically 4 is the unique identity of that particular cytochromal enzyme, and 3 is the family, A is the subfamily. So this is about the nomenclature of the cytochrome enzymes. Now let's discuss about the sites where the drug metabolism occurs. So there are various sites where the drug metabolism occurs. It includes the epithelial cells of GIT, in liver, in nasal mucosa, in lungs, and the drug metabolizing enzyme, that is cytochromal enzyme, then flavin containing monooxygenases, then epoxide hydrolysis. All these are the phase 1 enzymes, and UGT, that is UDP glucuronosyl transferase, which is a phase 2 enzyme, are present in endoplasmic reticulum. As you can see here, this is a magnified image of endoplasmic reticulum, and here you can see the cytochrome enzyme which is being projecting out from the endoplasmic reticulum, that is, it is projecting outside the lumen of the endoplasmic reticulum. So we have other enzymes like sulfotransferases, and then we have glutathione S transferases, which are present in the cytosol of the cell. Okay, so in cytosol we are having these two enzymes, which are basically the phase 2 enzymes, and the phase 1 enzymes are present on the endoplasmic reticulum. And apart from the phase 1 enzyme, one phase 2 enzyme which is also present in the endoplasmic reticulum is the UGT, that is UDP glucuronosyl transferase enzyme. Now for a drug to be given by once daily dosing, that is, in a day we are giving that drug for only once, it has to be eliminated within 24 hours. Because if the drug is not getting eliminated in 24 hours, then if we are giving the drug by OD dosing, then it can actually lead to the accumulation of the drugs. Either it can, if the elimination takes a time which is more than 24 hours, then it will lead to accumulation, and if it takes a time which is less than 24 hours, then it will lead to decrease in the effect of the drug, because it will be completely metabolized even before we are giving the next dose. So for a drug to be given by OD dosing, that is once daily dosing, it has to be metabolized and excreted out of the body within 24 hours. Now coming to the CYP superfamily of the drug metabolizing enzymes, so they metabolize the drugs and xenobiotics. So as I have already mentioned, drugs and xenobiotics, they both are metabolized by the cytochromal enzymes. They also metabolize and synthesize several endogenous compounds like the hormones like estrogen. Then we have bile acids also. So CYP19 is one group of this CYP superfamily which converts testosterone to estrogen. So this is a substrate specific cytochromal enzyme, that is, it acts only on this testosterone or androstenedione to produce estrogen. It is not going to act on other substrates. Whereas there are some other CYPs which also can act on different substrates. So these are called as substrate promiscuous, that is, they are unselective in nature and they have indiscriminate behavior towards the substrates. Because of this, they can metabolize several types of substrates, and this is possible only because they are having a large and a fluid substrate binding site. But because the activity is non-specific and the area is also large, so the substrate binding site means it is not that concentrated. Because of this, the metabolism can decrease, and this is leading to the decreased turnover in case of the CYPs which are not substrate specific and are substrate promiscuous. But still it can cause the amount of metabolism which is required by the individual. Next, coming to the CYP gene polymorphism. So by polymorphism we mean there is a presence of two or more variants form of a specific DNA sequence that can occur among different individuals or population. And one of the most common type of polymorphism is SNP, that is single nucleotide polymorphism. So single nucleotide polymorphism, it is responsible for causing inter-individual differences in the drug metabolism, and as a result this can lead to toxicity and efficacy, increased efficacy also. So we will not be expecting that this drug will be producing such an effect in such a low dose, or we will not be expecting that this drug will be producing such a toxic effect in such a low dose. But because of the variations in the gene which is responsible for coding these cytochromal enzymes, it leads to this variation in the drug response in between the individuals. Now we are also having several inhibitors and inducers of the cytochromal enzymes. So for example, we have CYP3A4. So CYP3A4 is stimulated by St. John's wort and it is inhibited by grapefruit. So it is not grapes, it is grapefruit. And Also, when we are giving two drugs which are metabolized by a particular single cytochromal enzyme, as in case we are giving ketoconazole and then we have protease inhibitors which are used for antiretroviral therapy, and here we are using it for antifungal therapy. So when these two drugs are given together, since they are metabolized by one particular group of cytochromal enzyme, that is CYP3A4, so as a result there will be competition between these two substrates for the enzyme, and as a result this will lead to decrease in the metabolism of either the ketoconazole group or the protease inhibitors, which will lead to toxicity. So we should always keep in mind enzymes which are responsible for metabolizing the particular drugs, and when two drugs are metabolized by a particular enzyme, then in that case we should be cautious about the fact that it can lead to toxicity of the either of the drugs when they are co-administered together. So this is the reason for several drug-drug interactions which is seen in case of the drugs metabolized by the CYP group of enzymes, because CYP group of enzymes can be easily induced, and CYP group of enzymes are involved in the metabolism of several drugs. Now coming to the other phase 1 enzymes, that is flavin containing monooxygenase. So in this we have 6 families. Out of that, FMO3 metabolize etopride, cimetidine and ranitidine, and it is the one which is abundant in liver. FMOs are actually considered as minor contributor to drug metabolism, and they also cause less drug to drug interaction, which is not the case in case of cytochromal enzymes, because they are not readily inhibited and not readily induced, unlike the cytochromal enzymes. Next we have the hydrolytic enzymes. Under that we have epoxide hydrolase. So epoxide hydrolase is responsible for metabolizing carbamazepine, that is it metabolizes the active form of the carbamazepine, and this enzyme is inhibited by valproate. Next we have carboxyl esterase. So carboxyl esterase metabolizes the drug, that is irinotecan, which is an anti-cancer drug, and these esterases are also less involved in the metabolism, just like this, unlike the cytochromal enzymes. So they are also not much involved in drug-drug interactions, just like the flavin containing monooxygenase. In this diagram you can see that irinotecan is converted into SN38. So SN38 is basically the active form of this irinotecan, and finally it is getting metabolized with the help of the enzyme, that is UGT1A1/7. So glucuronidation occurs, which finally converts it into SN38 glucuronide, which is the inactive form, and as a result it facilitates the elimination of this via bile. Now coming to the phase 2 reactions. So phase 2 reactions occur mainly in the cytosol, with an exception glucuronidation, which I have already mentioned, that is glucuronidation occurs on the luminal side of endoplasmic reticulum. UGT, that is the enzyme which is responsible for causing the glucuronidation, is present in highest concentration in the gastrointestinal tract and in liver. So what it does is it causes the formation of the glucuronide conjugate of the drug in the liver, and then this can be released into the plasma, and finally it can be excreted via urine, or else it can enter into the bile duct and it is excreted with the bile and it reaches the intestine. Whereas in intestine, that is basically in the large intestine, we have some of the microbes which are responsible for producing beta glucuronidase. So beta glucuronidase, what it does, it deconjugates the glucuronide from the drug, and as a result this drug is getting again reabsorbed and it reaches the liver. So it is called as enterohepatic cycling, and this is responsible for increasing the half-life of the drug, and the remaining drug, which is actually not getting converted to the active form again, will be excreted via feces. So this is the mechanism by which glucuronidation occurs and how it is being affected because of the presence of the microbes which are responsible for producing the beta glucuronidase enzyme in the large intestine. Now let's discuss a little bit about the UGT1 group of enzymes. So there are total 19 human genes which are encoding the UGT proteins, that is UGT enzymes. Out of that, 9 are encoded by UGT1. So as you can see in the UGT1 locus, we are having 9 different group of enzymes: UGT1A1, UGT1A3, UGT1A4, UGT1A5, UGT1A6, UGT1A7, UGT1A9, UGT1A10, UGT1A8. These are the 9 which is present in the UGT1 locus, and the rest 10 genes are actually encoded by the UGT2 family of genes. Okay, so this is about the UGT1 locus and UGT2 locus. So in case of UGT1 locus, we can see that exon 2 to 5 is common for almost all the genes, and whenever there is a change or a change in the sequence in this region, that is exons 2 to 5, it will lead to the change in effect of the enzymes, all the enzymes which are being produced by this locus. But if there is a change in exon 1 only, that is exon 1 region of the UGT1, then it will lead to the change in a particular enzyme only in which the defect is present. Now coming to one clinical implication, so we know that this UGT1A1 does the glucuronidation of bilirubin, and this is one of the rate limiting step for bilirubin clearance of the body, which is actually essential for the body. So if it is affected, it can lead to several syndromes. We have several syndromes like Crigler-Najjar syndrome type 1 and type 2, and we have Gilbert syndrome. So in case of Crigler-Najjar syndrome type 1 and type 2, we have the problem in UGT1A1 gene exon. So here we have the issue, that is the issue is present in the exon region in case of Crigler-Najjar syndrome, whereas in case of Gilbert syndrome there is problem in the promoter region. So the promoter region is actually not the one which is usually present in the individual, so this leads to Gilbert syndrome, and as a result both of these will cause hyperbilirubinemia. In Crigler-Najjar syndrome type 1, there is complete lack of bilirubin glucuronidation. So in that case, we cannot actually do much in the treatment. But in case of Crigler-Najjar type 2, it is actually a comparatively less severe variant of the Crigler-Najjar syndrome. So here, in case of Crigler-Najjar type 2, that is Crigler-Najjar syndrome type 2, we can actually use the agents that induce UGT1A1 gene expression, such as phenobarbital. So we can use the drugs like phenobarbital, which is responsible for inducing the UGT1A1 enzyme for the management of Crigler-Najjar syndrome type 2. Now, so this is basically the variants which are responsible for causing the Gilbert syndrome. As you can see here, UGT1A1 star 28. So this star 28 basically means the star allele which is responsible for causing the variation, that is responsible for causing Gilbert syndrome. Then we have other substrates like which are actually the substrate of UGT1A1. It includes irinotecan, then we have atorvastatin, then we have gemfibrocil, then we have etanyl estradiol, then we have ibuprofen. But atazanavir is also mentioned here, but atazanavir is not a substrate, but it is actually an inhibitor of UGT1A1 function, that is not a substrate for UGT1A1. And here you can see the metabolism of SN38G, that is SN38G, that is basically the irinotecan metabolism and how it is being affected. Now coming to the next group of phase 2 reactions, that is sulfation. So sulfation is done with the help of sulfose transferase enzyme. So sulfose transferase enzyme plays a major role in the normal human homeostasis. SULT2B1B is expressed in the skin and it is responsible for causing the catalysis of cholesterol. And SULT2A1 is highly expressed in the fetal adrenal gland where it produces glutathione S transferase. So it leads to the formation of XGSH conjugate, that is the substrate and that is the drug and the glutathione conjugate. So one drug which is very important and needs to be remembered in case of glutathione conjugation is the example of paracetamol, that is acetaminophen. So acetaminophen is converted to its toxic metabolite, that is NAPQI, that is N-acetyl P-benzoquinone imine. So NAPQI, so it is actually conjugated with the help of glutathione, as you can see here. So in case of normal dosing of acetaminophen, it is easily managed, that is the toxic metabolite is easily conjugated with the help of glutathione, and finally it leads to the excretion of that toxic metabolite. But in case there is an overdose of acetaminophen, then in that case it will lead to depletion of the glutathione. So for that case we are giving one drug, that is N-acetylcysteine, which is actually the antidote for paracetamol poisoning because it replenishes the depleted GSH level, and as a result it helps in detoxifying the NAPQI, that is the NAPQI, which is the toxic metabolite of the acetaminophen, and hence preventing the toxic effects of NAPQI in the cells and preventing the hepatic necrosis, because mainly this NAPQI is causing the liver damage. Now coming to the rest of the phase 2 reaction, so we have acetylation, which is catalyzed by NAT, that is N-acetyl transferase. So we have several genes, that is NAT1 and NAT2. We can have slow and fast acetylators of the drugs. So the drugs which are being metabolized by acetylation includes isoniazid, which is the anti-tubercular drug. Then we have hydralazine, then we have procainamide, then we have dapsone. So if the isoniazid is not metabolized properly, then it will lead to the adverse effects like peripheral neuritis and hepatotoxicity. Then next, if hydrolyzine is actually metabolized slowly, then it can lead to several other adverse effects like hypotension, that is decrease in BP. Then we have sympathetic baroreceptor reflex. Then procainamide, when it is not metabolized properly, it again can cause hypotension. Then it can also cause bradycardia. And dapsone can cause hemolysis if it is not metabolized properly, hemolysis and methemoglobinemia. So here you can see isoniazid, which is actually undergoing the phase 2 reaction, that is with the help of N-acetyl transferase, and it is being converted into acetyl isoniazid. So here you can see the addition of the acetyl group to isoniazid. Now coming to the next phase 2 reaction, that is methylation, which is catalyzed by the enzymes like COMT, that is catechol O methyl transferase, then POMT, phenol O methyl transferase, then we have TPMT, that is thiopurine methyl transferase, and then we have TMT, that is thiol methyl transferase. So for methylation, we need the methyl group. Methyl group is to be added to the drug. So this methyl group is being donated by the molecule, that is SAM, that is S adenosyl methionine. S-adenosyl methionine. So S-adenosyl methionine is basically the methyl donor which provides the methyl group for the methylation reaction. So the different enzymes which I have mentioned here, that is COM, that is catechol O methyl transferase, is responsible for metabolizing methyl dopa, dopamine and norepinephrine. Then TPMT, that is thiopurine methyl transferase, is responsible for metabolizing the drugs like azathioprine, then 6-mercaptopurine and thioguanine. So in case of deficiency of TPMT, it leads to severe toxicity. This is because these drugs are anti-cancer drugs and they have several severe toxic effects. So because of this, we need to do the genotyping so as to look for the presence of inactive TPMT alleles, and as a result, we will then decrease the dose of these anti-cancer drugs in the individuals, because in that way we can easily prevent the toxic effects. Now coming to the significance of metabolism in drug development. So an ideal drug candidate would be metabolized by several CYPs, and as a result it will be having less drug interaction because it is metabolized by several CYPs. So even if one CYP is not functional in the human body, then it can be metabolized by the other CYPs. So we have several computer-based computational in silico studies which are actually responsible for developing the drugs. So how the computational methods helps in developing new drugs is, that is basically it does the prediction of the drug metabolism. So the structures of several CYPs have been determined with the help of this method, including the CYPs like CYP2A6, CYP2C9, CYP3A4. So these structures may be used to predict the metabolism of a drug candidate by fitting the compound to the enzyme's active site, which is responsible for causing the particular metabolism. Then we are also using metabolomics. Metabolomics are used to screen for the drug toxicity in the whole animal systems during the preclinical drug development, and because of this it has decreased the need for necropsies. There is one particular disadvantage with this computer based, that is in silico studies, that is the structures which are determined by X-ray analysis of the crystals of the enzyme substrate complex are actually static in nature, but we know that enzymes are flexible, and as a result this is one of the limitation of these computer based studies for the development of the new drugs. We can use several animal models also, like we can use the humanized mice, then we can also use human liver transplantation in an immunocompromised mice. So basically this will make the mice more human in nature, that is it will introduce the human character into the mice. That is, if we give the human liver replacement in the immunocompromised mice, then the liver will not be rejected, first graft rejection will not occur, and then we can see the action of the liver, human liver cells on different drugs. Or else we can make the mice humanized. So what is this humanized mice? Humanized mice is basically a genetically modified mouse that has the functioning human genes, cells, tissues, and or organs. And humanized mice are commonly used as small animal models in biological and medical research for human therapeutics. So these are the two things which we can do. That is humanized mice, and next we can do the human liver replacement in the immunocompromised mice. Now here you can see several articles. So you can see one article from the Journal of in silico and in vitro pharmacology, which are supporting the role of pharmacogenomics in drug discovery and development. Then we have role of pharmacogenomics in drug discovery and development from other articles also. This is the one which is being produced by some of the authors from the Department of Pharmacology, JIPMER in India also. So we can see several studies where we are actually stressing upon the fact of pharmacogenetics and considering it for the purpose of drug development. Next, coming to the therapeutic implications of pharmacogenetics. So, drug metabolism, drug transport, drug receptors, and the non-targeted areas can be affected by the presence of different pharmacogenes. This is already being described in the previous slides. So, these pharmacogenes, that is important variants in pharmacogenes, can be so rare that it can be documented only in a single individual. That is so rare that it can be documented in a single individual, and it can be so common that it is just like the wild type. So but what do I mean by wild type? Wild type is basically the normal type of that particular pharmacogene. Okay, so we can have different types of variation. That is, we can have single nucleotide variation, then we can have copy number variation. So in single nucleotide variation, there will be change only in one single nucleotide, and it is relatively easy to identify and analyze, and it has a smaller impact on the human phenotype because only one single nucleotide is changed. Next, in case of copy number variation, it can lead to either gene deletion. As you can see here, the gene A, which was present in the normal individuals, is absent here, or it can also lead to gene duplication. That is, there are two gene A, that is gene A and gene A plus in the new individual. So this is actually difficult to identify and analyze, but it is also responsible for causing a greater impact on the human phenotype. Means the resulting phenotype will be very much different here, because here a complete group of nucleotides, not a single nucleotide but a complete group of nucleotides, a single gene is actually affected here. Coming to the term linkage disequilibrium. So what do we mean by linkage disequilibrium? So whenever genetic variants are inherited, they are actually not inherited in isolation, rather they are inherited as constellation, that is a group of variants which are found together, and this is what we call as linkage disequilibrium. Then coming to the star allele nomenclature. So as you can see here, we are having several cytochrome enzymes. So the enzyme which we are going to study, that is the gene which is responsible for coding that particular cytochrome enzyme, is CYP2C19. Okay, so first we have CYP2C19 star 1. Star 1 is the star allele, and it is actually the wild type, so it will be having normal effect. Then we have CYP2C19 star 2 and CYP2C19 star 3. So in this we have a mutation, that is the star allele here is 2 and 3, and because of this it will lead to no function of that particular enzyme which is coded by this gene. Then we have CYP2C19 star 17. So here the mutation is represented by 17. So if 17 is present, then it will lead to increased expression of that particular gene, leading to formation of a more active metabolizing cytochromal enzymes. So we have different combinations. That is, when we have a combination of CYP2C19 star 17, both of the alleles are actually responsible for increased expression. Then dehydroepiandrosterone sulfate, which is required for placental estrogen biosynthesis. Next, we have the copy number polymorphisms within the SALT1A1 and SALT1A1 and SALT1A3 and SALT1A4 genes, which are responsible for causing the inter-individual variation in the expression and activity of these enzymes. Copy number variants is comparatively bigger than the single nucleotide polymorphism. That is, here actually a large group of genes are actually altered in the final gene product. Next, we have glutathione conjugation. So glutathione conjugation is basically conjugating the substrate of the drug with the help of glutathione, and it is catalyzed by the enzyme that is GST, that is then it will lead to the formation of the enzymes which are ultra rapid metabolizer, that is these individuals will come under the category of ultra rapid metabolizer. Then in case one allele is normal and the other one is highly active, then it will lead to the formation of rapid metabolizer group. Then both the alleles are normal, in that case it will be normal metabolizer. One allele is normal and other allele is inactive, then in that case it will form intermediate metabolizer group. And if both the alleles are inactive, then it will lead to the formation of poor metabolizer group. So we have total 5 groups: ultra rapid metabolizer, normal metabolizer, intermediate metabolizer, and poor metabolizer. This is based on the different permutation and combination of the alleles, and the alleles varies because of the mutation. Now here you can see the different CYP2C19 phenotypes in different population, that is sub-Saharan African population, then Oceanian, European, East Asian population, then we have African American population. So in different population we have different varieties, that is ultra rapid metabolizers are different than in number in case of all the different populations. Now coming to another example, like let us take one example, that is tacrolimus, which is actually metabolized by the CYP enzyme, that is CYP3A4 5. So if we have CYP3A4 star 1 oblique star 1, in that case it is having high metabolizing function compared to star 1 oblique star 3, compared to star 3 oblique star 3, that is least active. So in the first one, when we are giving the tacrolimus, it will lead to the metabolism of tacrolimus, and as a result there will be the decreased concentration of tacrolimus, whereas in the third group there will be maximum concentration of tacrolimus. So because of this, in first group, that is CYP3A5 star 1 oblique star 1, we have to give an increased dose of tacrolimus compared to that of CYP3A4 star 3 oblique star 3, where we should decrease the dose of tacrolimus because here the metabolism is not occurring properly. So this is the implication of the pharmacogenetics and the pharmacogenomics in the drug metabolism, and how we need to monitor the dose and how we need to actually modulate the dose for different types of individuals based on their genetic makeup. So here you can see the list of several selected well-established clinically actionable pharmacogenes, that is CYP2B6 is seen in case of efavirenz, that is it is metabolizing efavirenz. Then we have CYP2C19 involved in the metabolism of amitriptyline, citalopram, clopidogrel, escitalopram. Then we have voriconazole, pantoprazole, omeprazole, and we have several other enzymes, that is CYP2C9 for the metabolism of fosphenytoin, ibuprofen. Then we have phenytoin, warfarin. So they affect the drug efficacy, toxicity, and they are hence going to change the amount of the dose which is required for producing the pharmacological effect. Then we have CYP2D6. Then we have CYP3A4, which is responsible for the metabolism of tacrolimus. Then CYP4F2 for warfarin. Then we have other enzymes like DPYD. Then SLC01B1 for simvastatin. Then we have modifiers, that is G6PD, that is it is not going to actually affect the pharmacological effect, but it is going to affect the other effects of the drug, like the some of the adverse effects like G6PD deficiency and all, can actually lead to several toxic effects of the particular drug, that is G6PD. In case of raspberry case, tafenoquine is the antimalarial drug, so it can lead to several risk of toxicities like hemolytic anemia, methemoglobinemia. Then we have UGT1A1, that is it is actually affecting irinotecan metabolism, so it can lead to risk of toxicity if it is not actually working properly. Then there can be actually competition between bilirubin and irinotecan, and this can lead to decreased metabolism of bilirubin, leading to the condition of hyperbilirubinemia. Now we have an example of a drug, that is warfarin, which is actually affected by several genes. So this is what we call as multigenic pharmacogenomic trait. That is, multiple genes are responsible for actually affecting the function of that particular drug and effects of that particular drug. So warfarin, as you can see, is primarily metabolized by CYP2C9 to its inactive metabolite, that is 7-hydroxywarfarin, and it is also metabolized by some other CYPs also. Next, warfarin actually is exerting its anticoagulant effect by inhibition of VKORC1. That is, VKORC1 is an enzyme which is necessary for the reduction of the inactive form of the vitamin K to active form, that is the vitamin K which is the reduced form. As a result, when we are inhibiting VKORC, which is actually causing the conversion of the inactive form of vitamin K to the active form, we are again causing the anticoagulant effect. Plus, this active form of vitamin K, that is vitamin K reduced, is converted to its inactive form by another enzyme, that is CYP4F2. So here you can see totally we are having three genes which are actually responsible for affecting the function of warfarin, that is CYP2C9, then we have VKORC1, then we have CYP4F2. So these three are going to affect the pharmacological properties of warfarin, and this is a classic example of multigenic pharmacogenomic trait. Now coming to the process of DNA extraction, this is because how can we study the genes? First we have to actually isolate the genes, that is the DNA and all, and then only we can study the different composition of the genes. So for this what we do is first we take the sample, that is the biological sample. It can be blood or it can be any other tissue. Then we do the lysis of the sample so that the DNA comes out of the cells. Then the capturing and clearing of the DNA is done, and finally the elution of the DNA, that is we can see the different sequence of the nucleotides. So this is the process of DNA extraction in short. Actually the detailed process is to be mentioned, but that can be explained in a complete different presentation. So this is about the DNA extraction. Now coming to the different methods of pharmacogenomic discovery. So under this we are having two approaches, that is the candidate gene approach and then we have the gene agnostic approach. Okay, so what are the difference between candidate gene approach and the gene agnostic approach? So a candidate gene approach basically requires a prior hypothesis. Okay, a prior hypothesis that particular genes of interest contain the variants that may be associated with a trait or a disease. So as you can see here, first we do the in vitro studies or the animal studies, then we select particular subset of the candidate disease genes which are actually responsible for causing a particular human disease. So the limitation is the knowledge of the genes which we have. Whereas in case of gene agnostic approach, what we are going to do is it is a method of analyzing the whole exome and the whole genome of the individual with no panels or other perceptions. That is, like, there will be no previous perceptions about the presence of the variants and the location of the variants which are responsible for causing that particular condition. So the limitation here is we are going to take a lot of time and a vast amount of variants will be detected, and we need effective strategy to filter and prioritize those variants. So that is, first we analyze the patients to identify the novel human disease genes, then the genes with unknown function or new function identified for the particular gene is being seen in case of in vitro studies and in vivo studies in animal models. So gene agnostic approach is totally opposite of the candidate gene approach. A genome-wide association study, that is GWAS, is an approach to compare the genomes from many different people to find genetic markers which are associated with a particular phenotype or risk of disease. So genome-wide association study is basically going to come under this approach, that is the gene agnostic approach. So this is about the different methods of pharmacogenomic discovery. Finally coming to the conclusion of this seminar. So as you can see, when a particular medication is given to the individuals, it can produce no response, it can produce the desired response, it can produce serious side effects with the same dose. So it means there is inter-individual variation in the drug response for a particular dose of drug, and this can be due to several attributes, one major attribute being the genetic makeup. That is the reason why we need to do the pharmacogenetic testing for the individuals, but it is costly, but it can still be actually implemented. So what it is going to do is, first the physician is actually going to order a pharmacogenetic test, then the sampling of the biomaterial occurs, then it is being sent to the laboratories, then we analyze data and generate report, then we review the test results with the patient, and this leads to the development of a personalized dosage regimen for the particular individual, because all the individuals will be having different genetic makeup leading to different dosage requirements. So this is the implication of pharmacogenomics and pharmacogenetics in the individualization of the dosage regimen for the individuals. So these are the references which I have referred to for creating the slides. So with this, I would like to end this presentation. So I hope you like this content. So if you like my content, please do like, share, subscribe, comment, and also press the bell icon so as to get notified whenever I upload a new video. Thank you guys.